On behalf of APJ Abdul Kalam Technical University, Lucknow, as well as Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida, I would like to extend a very warm welcome to all of you for my class today on Pharmaceutical Microbiology, which is a subject which you are studying in your B.Pharm third semester. This subject is covered across the country as recommended by Pharmacy Council of India, and we are in our university also you are studying this subject. Now, today my class will be mainly covering the microbiological assay of vitamins and amino acids. Now, in our previous classes we have discussed about how these microorganisms are beneficial not only for the human beings, as well as they are beneficial for the pharmaceutical industries also. The pharma industries have taken the advantage of these microorganisms in evaluating or estimating the quality of the antibiotics, where we estimated or measured the inhibitory capacity of the antibiotic to inhibit the microbes. But this microbiological assay of vitamins and amino acids is a little bit different in the sense that in this case, instead of measuring the degree of inhibition, we are measuring the degree of exhibition. That is, these vitamins and amino acids are essential for the growth and multiplication of the microorganisms. Now, that is the reason why without these vitamins or without these amino acids, certain microorganisms cannot grow or multiply. In fact, their growth is so critically dependent on these vitamins and amino acids that more is the vitamin, more is the growth, lesser is the vitamin, lesser is their growth. So, what we estimate is, we estimate the degree to which they are growing, the degree to which they are producing the metabolite, and linking their growth with the concentrations of the vitamins and amino acids. So, keeping that perspective in our mind, where we have to measure the degree of exhibition, vitamin assay can be estimated or assayed by two techniques. One is called Turbidimetric method and the other one is called Titrimetric method. Now, we will be studying in our class today by taking one vitamin which we require in our daily life. Here today we will be studying, we will be taking vitamin B12. We will be starting with respect to vitamin B12 and see how the microorganisms help in estimating the amount of vitamin B12 present in a particular pharmaceutical preparations. As I have told you that there are two methods, one is called Turbidimetric method and other one is called Titrimetric method. So, first we will be starting with the Turbidimetric method. Now, let us first study what is the principle. The basis of this assay is to measure the ability of the test organism to utilize the substance being assayed under proper nutritional conditions. We should select the organisms in such a manner that its growth must be dependent on that particular vitamin which you are wanting to assay. That is, that organism should be growing and the growth should be vitamin concentration dependent. So, what we measure is, we measure the response, that is the growth of the organism is proportional to the dose or the amount of the factor which is added to the media. That is, the growth is directly proportional to the concentration of the vitamins. Similarly, will be the case for amino acids. The principle remains the same. What we have to prepare is, initially you have to prepare a media which you are calling as the basal media. The basal media is such that it will provide all sorts of nutrition, but even after getting all sorts of nutrition, that particular microorganism will fail to grow because we are not adding vitamin to it. So, we have to prepare a basal media for a particular organism. Say for example, in case of vitamin B12, we have to select Lactobacillus leishmani. This particular organism will not grow in this basal media unless you are adding vitamin B12. You see, in this particular slide, we are having cysteine, tryptic digest, casein acid hydrolyse, we are having glycine, cysteine, asparagine and so on. The entire table having a wide range of compounds which are by themselves supporting the growth of the microbes. But the microorganism Lactobacillus leishmani will not grow unless you are adding vitamin B12 in this particular media. And we have to prepare this basal media, sterilize it, and the pH has to be maintained to 6.8. So, let us see what are the prerequisites to the assay. The test organism selected must be able to utilize the free cyanocobalamin. Lactobacillus leishmani, as I have told you, is the organism which we have to use. That is, this Lactobacillus leishmani growth is very crucially dependent on the concentration of vitamin B12. This organism Lactobacillus leishmani is selected as because it is very easily available, it is non-pathogenic in nature, it is easily culturable, mainly obtained from milk, cheese and other dairy products. Due to easy availability, non-pathogenicity, easy culturability, easy maintenance, Lactobacillus leishmani is selected to test or determine the concentration of vitamin B12. So, we have to prepare an inoculum. Inoculum means we have to prepare a media, a sterile culture media, where this Lactobacillus leishmani should be growing. and we have to make repeated subculturing, keeping the temperature at 37 degrees Celsius, and generally the time is 18 to 24 hours. So, what we have to do is we have to prepare a standard stock solution where the concentration is 1 microgram per ml, and we have to use 25% ethanol. And from this stock solution, we have to prepare a series of varying different concentrations of standard vitamin B12. We have to prepare from this stock solution a series of varying concentration of vitamin B12, and those concentrations should range from 0.01 to 0.04 microgram per ml. This table shows that we have to take test tube number 1 to test tube number 10. All these are having standard solution of vitamin B12, and remaining 4 we are having test or sample solutions of vitamin B12. We have to add 5 ml of basal media. The total volume needs to be 10 ml. In test tube number 1, we need not add any standard solution, 0 ml. Test tube number 2, 0.5 ml of standard cyanocobalamin solution, 1.5, 2, 2.5, 3, 3.5, 3, 4, 4.55. These are the 10 test tubes containing standard cyanocobalamin, and that too in this volume. 5 ml media standard to all the test tubes. As because the total volume is 10 ml, you have to make it up with the help of sterile distilled water. Apply the same process for test solution, where you take 1 ml, 2 ml, 3 ml, 4 ml of the test solution, add the same quantity of the basal media. Remember, the basal media is such a media where the microbes will not grow unless vitamins are added, and the volume again has to be made up to 10 ml capacity. So, that is what we have written here. Fill 10 test tubes for standard cyanocobalamin solution, 5 ml basal free media, volume to be 10 ml, then they are to be sterilized. After adding this microorganism, we have to incubate all these test tube to 30 to 37 degrees Celsius for a period of 16 to 24 hours. Then we have to measure the turbidity created by measuring the optical density, taking the help of UV visible spectrophotometer, keeping the wavelength at 640 nanometer. So, you will be getting a standard graph, where optical density versus concentration, and you determine, so that will be a standard graph of optical density versus concentration as prepared with the help of optical densities and concentration of the standard solutions of vitamin B12. You are the unknown also, you determine the optical density, straight away put it on that particular graph, the point where it cuts the curve, drop a line which is parallel to the y axis, where it cuts the x axis will give you the concentration of the unknown sample of vitamin B12. Next, we are going to the next method, which we are calling as a titrimetric method, where we are doing a process of titration. Clean all the 10 test tubes and add 10, 1, 1.5, 3, the way we did exactly in the same way. We have to add a varying vitamin standard solution. To each, we have to add a fixed quantity 5 ml of the basal media, make up the volume up to 10 ml with sterile water. Similarly, take additional 4 test tubes where we are adding the test solution to be added. Add 5 ml of basal media, again make up the volume with 10 ml as told earlier. This is the process which we adopt for titrimetric method also. Again, this is for the standard, this is for the unknown or the sample. This is the concentrations 0.5, 1. And fixed volume of the basal media to be added, volume made up to 10 ml with the help of sterile water. So, sterilize all the test tubes in an autoclave at 121 degrees Celsius for 15 minutes at 15 pounds per square inch. After sterilizing, cool the test tubes, inoculate with a drop of Lactobacillus leishmani, which is overnight inoculated. We have to add Lactobacillus leishmani. This is the microorganism we have to add to all the 10 test tubes containing standard solution, as well as we have to add it in the 4 test solutions containing test tubes. Now, after you are adding this inoculum of Lactobacillus leishmani, then you allow it to grow by providing a favorable atmosphere in an incubator. So, you have to put all these test tubes in an incubator for 64 to 72 hours, 64 to 72 hours, maintaining a temperature of 30 to 37 degree Celsius. Now, when they are growing, automatically they are producing their metabolites, and after the incubation process is over, that is after 72 hours, after the incubation process is over, we have to titrate the content of each test tubes against 0.05 normal sodium hydroxide using 0.1% weight by volume bromothymol blue as an indicator. I am repeating once again. Whenever you are adding or all this basal media and the standard solutions and the organism will try to grow because there is a presence of vitamin B12 during the process of incubation. Now, after incubation, you have to take out those test tubes and titrate the content of each test tubes with 0.05 normal sodium hydroxide using 0.1% weight by volume of bromothymol blue indicator, and at the end point there will be a change of color to green. At the end point there will be a change of color to green. So, you have to determine the average titration value for each of the standard as well as the test sample. You have to determine the titration value for each of the standard as well as for the test sample whose concentration you want to measure, whose concentration you want to assay. Then you have to plot, then you have to plot the titration value against the concentration of the standard solution. That is, you have to plot the titration value which you are getting and the concentration of the vitamin standard solution. So, you will be getting a graph, then you take your unknown one or the sample one, you will be getting a corresponding titration value. Simply put the titration value in the graph and the point where it cuts the curve, the point where it's curved the graph, go for the process of extrapolation by simply drawing a line parallel to your y axis. Wherever it will be cutting your x axis will give you the concentration of the unknown sample. Wherever it will be cutting the x axis, that will be giving the concentration of the unknown cyanocobalamin solution. So, let us come to the interpretation of the results. Determine the average titration value for the standard as well as the solutions. Plot the graph considering the average titration value in ml of 0.05 normal sodium hydroxide against concentration of the standard cyanocobalamin solution. So, as told earlier that a graph will be resulting, the graph will be prepared with the standard vitamin B12 solution, and then by the process of extrapolation a standard curve is obtained, and you have to determine from the graph the concentration of the unknown sample of cyanocobalamin is determined. This is it basically looks like, that is titration value on the y axis, standard cyanocobalamin solution concentration on the x axis. Say this is the value of the unknown, this is the value of titration value of the unknown, titration value of unknown or sample. Straight away draw a straight line, the point where it is cutting this graph, draw a straight line, and wherever it is touching this x axis, that will give you the standard concentration, that will give you the concentration of the unknown sample. This is basically a pictorial representation of the turbidimetric method, the way we discussed it just now. Next, we are moving on to the assay of amino acids. Now, there are various amino acids like valine, leucine and methionine. We can be easily assayed with the help of these microorganisms. So, again we have to prepare another media which is called Bacto amino acid assay media, where we had all the ingredients except the amino acid which we want to test. We will add all the ingredients except the amino acid which we want to test. So, in all the other test tubes, add graded concentration of standard amino acid. Similarly, you have to take another test tubes where we will be, instead of standard, you will be taking the sample. The sample you have to calculate, the sample we have to determine. Now, all these test tubes needs to be prepared in triplicate. Why triplicate? Because the error will be minimized. And then we have to add 2 ml of amino acid media, which you are calling as the Bacto amino acid media, to all the test tubes. So, what we have done? We have added in the test tubes either standard or we have added the sample, and to all the test tubes we have added 2 ml of the amino acid assay media, which you are calling as the Bacto amino acid assay media. And then all these test tubes are autoclave under standard solutions, standard conditions. Then you have to add the appropriate organism. Now, this organism is again amino acid specific. This organism is again amino acid specific, that is Streptococcus faecalis ATCC 8043 is to be added whenever we are estimating either valine or leucine as amino acid. We have to add leuconostoc mesenteroids ATCC 8043 whenever we are going for the assay of methionine. I am repeating once again. In case of vitamin B12, you have taken Lactobacillus leishmani. But in case of amino acid, the organism is changing. Why? Because there you have taken vitamin B12, here you are taking some other amino acids. Now, the amino acid, if it is valine or leucine, you have to take Streptococcus faecalis ATCC 8043. Whenever we are adding methionine, you have to take leuconostoc mesenteroids ATCC 8043. This you have to add in all the test tubes. Then incubate it at 37 degrees Celsius for a period of 17 days and then measure the optical density at 600 nanometer. You have to measure the optical densities for all the test tubes at 600 nanometer. Now, the optical density so obtained is to be subtracted from the blank. You have taken one test tube where you have not added any vitamins or you have not added any amino acid. That was the test tube which was known as blank. So, that optical density you have to subtract from all the test tubes which are containing varying dilutions of standard amino acids, also for test amino acids. So, you have to determine the optical density so obtained are subtracted from the blank and are plotted against the standard amino acid concentrations. And here standard amino acid concentration is taken on the x axis, optical density on the y axis, and then you have to determine the optical density of the unknown sample of the amino acid. Just as taught earlier, you have to extrapolate with the help of graph. You have to extrapolate the graph in order to get the concentration of the unknown sample of amino acids. Now, this is a process where these amino acids as well as vitamins, they are wonderfully estimated with the help of the microorganisms, and it has been seen that the only difference with the antibiotic and this amino acid vitamin assay is, I am once again giving you a bit reminder, a bit of recapitulation. Amino acids and vitamins, you are measuring the degree of exhibition, how they multiply, you are measuring the degree of exhibition. But in the case of antibiotics, which we have studied earlier, we have measured the degree of inhibition. So, in one we are measuring the degree of inhibition, in the other we are measuring the degree of exhibition. Both you are taking the help of the microorganisms. Therefore, microorganisms have been successful enough to evaluate the concentration of both the antibiotics as well as vitamins as well as amino acids. And I would like to request all of you to study this particular topic of vitamin assay and amino acid mainly from the Indian pharmacopoeia. With this, I am coming to the end of my class for today. Thank you very much for your patient hearing.